That's the gateway system. So, let's see if we can have a better picture of the different. Yeah, okay, this looks a little better. So remember, we have the at L, the at R, the at P, the at V. We're just going to abbreviate these to L, R, and PB, and so forth. And so here, if you'll notice, it's kind of hard to see, but we have the L1, the L2, and then we have the P1, the P2, the B1, the B2. You'd have to look at this a little more closely, and I'm not going to hold you accountable for that. But we go back and forth between the L versions, or the R versions, and these P or B versions, using the same system we saw before. Those certain enzymes that allowed us to go from POP and BOB to POB and BOP. Remember, we use those special, use 2 enzymes to go in one direction, and then 3 enzymes to go in the other direction. Well, they call that LR clonase. That's the proprietary name they give. And depending on which sort of enzymes you add, you can go in this direction and then start with just these guys and create the entry clone again. Or you can go from the entry clone and create these guys back again. So you can go back and forth between these things without using any restriction enzymes. All you use is the clonase enzymes that they give to you. Why would it be good for us to avoid using restriction enzymes and just start off with one entry clone, and then use the at L, at R, at P, at B systems and the enzymes to go from one entry clone to all the other ones? What would be the advantage? No restriction enzymes. Why is that good? What if you were creating a big library with very large pieces in it, and you wanted to put these, and you just had a giant library, and you wanted to be able to test a whole bunch of these in protein expression systems to see which protein is the most active?
You don't have to worry about restriction sites in the piece that you're cloning.
That's right. You don't have to worry about restriction sites in the piece you're cloning. So if we're cloning in 2 kb or 4 kb pieces, well, gee, you could easily have a restriction site in there. And if you very carefully plan to use 2 restriction enzymes, maybe, if you're looking at 100 of these things, there's a good chance that your restriction site that you chose will be in the middle of many of these. So now we can go ahead and just, with perfect freedom, be able to look at tens or even hundreds of these and be able to put them into all our vectors and not have to worry about it at all, those restriction sites. Yeah, exactly. Yeah. So that's the big advantage to it. What's the big advantage to the company? Well, they pretty much have you at a stranglehold. And so just to sell you these enzymes and the cloning kit, it winds up being hundreds and hundreds, if not thousands of dollars. So you better have a pretty big grant to use the gateway system. So otherwise, the rest of us peons are just using restriction enzymes and planning things carefully. So that's the only drawback to it. It shouldn't cost that much money, but it does. All right. And it makes things go more quickly, because you just have very easy, you don't have to think about which restriction sites you're using. You don't have to plan. You just pop it in the entry vector and away you go. All right, let's move over now to plant virus vectors. So we're going to do protein expression again, and we'll talk about TMV first. Tobacco mosaic virus. I'm just going to pop in TMV viral vector. Okay, we're going to look at this TMV-based vector. And as you've seen many times before, what this has is, in this case, it has a T7 promoter, in this particular case. And that means that it can be, we can take this DNA that we have, and we can add to the DNA T7 RNA polymerase that we buy from a company. We're actually going to add the protein. And then that protein will recognize this promoter, and it will produce an RNA corresponding to the TMV sequence. And then we have a linearization site. That's where we actually cut the TMV vector with a restriction enzyme. So it goes all the way to the end, and then in a rather primitive way, it just stops because we've run out of DNA. We've made a restriction enzyme cut in our plasmid. So we produce an RNA, which is exactly like the TMV virus. Why do we want to produce an RNA? Why not just use the DNA to cause an infection? Why do we go to the trouble? This is called in vitro transcription. Why do we go to the trouble of producing an RNA?
Is it going to just make straight protein with 2 infects?
Yeah, it's an RNA virus. I guess I asked too hard of a question. Sorry. It is an RNA virus. So you have this, you can't, the only way you can clone RNA is to actually put it in DNA and make it as an E. coli plasmid. They do the same thing with polio virus. That's why I brought this up, because it's similar to the animal virus vectors. It's just with the plant virus vectors you can make gobs of protein, so that's why they're useful for protein expression. So what you do is you go ahead and take the DNA, put it into a tube, add your T7 RNA polymerase. First you cut it. You have the linearization site right here. Then you make your T7 RNA polymerase, and you add it. And we have in vitro transcription. You take the RNA and you can rub it right onto a plant, believe it or not, and you get an infection. So you put your gene of interest right here at the multiple cloning site, MCS for multiple cloning site, and away you go. You can get lots and lots of virus. Now there are more sophisticated ways to doing this. Instead of a T7 promoter, let's put something called a 35S promoter. A 35S promoter. That's from a virus, as you can guess. We're always using virus promoters, called cauliflower mosaic virus. So similar to cytomegalovirus, it's abbreviated CAMV rather than CMV. But that's the, just as CMV is the go-to constitutive promoter for mammalian cells, CAMV is the go-to constitutive promoter for plant cells, cauliflower mosaic virus. And we use the 35S, which is the big RNA in cauliflower mosaic virus. It just happened. There's the 35S and the 19S. We use the 35S. That's the big RNA promoter. And so if we do that, we can actually, believe it or not, rub the DNA onto plants. It will, in some cases, get into the nucleus of the plant cells, and the 35S promoter becomes active, and the RNA pops out. So you can actually rub DNA. But there's something even better that we can do, and we'll talk about it quite extensively later. And so I'm not going to go into it much now. But there's a bacterium called Agrobacterium tumefaciens. I'll show it to you just real quick, just as a little teaser, I guess. But Agrobacterium tumefaciens is a fantastic and Agrobacterium tumefaciens causes a cancer in plants. And they used to get a lot of cancer money for this. Look at the size of that cancer. That's a big cancer. You wouldn't want that on you, would you? But if you're a tree, it's okay. And then there is also Agrobacterium rhizogenes, which causes root cancers. And we call these galls in the plant world. But Agrobacterium, what it does is this. It's just unbelievable. It happens only once in biology with the Agrobacterium. In all of life, it happens nowhere else. This is not, there we go. And what it does is Agrobacterium is able to saunter up to a plant cell, and it has something called a TI plasmid, tumor induction plasmid. This is the thing that causes the tumors. And it has something called tDNA. It transfers the tDNA, it just excises, and then using a very primitive secretion system, which goes way back in the lineage of life, it secretes to the exterior. It secretes the DNA. Normally it secretes protein, but this system has been hijacked by Agrobacterium to secrete DNA. Furthermore, along with that tDNA travels some proteins, and those proteins manage to get it into the cell, into the plant nucleus, and then, believe it or not, it inserts into the plant nucleus just like a retrovirus. So it has a left and a right border, and see these little red marks here? You know, in retroviruses you've got LTRs. Here, these little red borders we call left border, right border. The left border and right border are recognized by the proteins and inserts right into the plant chromosome. So if you take Agrobacterium tumefaciens and put it next to a plant cell, by golly, it's going to transform that plant cell like that. It's just incredible. If you had this in humans, you could really make rapid progress, but it's in plants. So it actually can work in mammalian cells, but I guess it hasn't been used that much. So what you can do, this is the magic. Replace all the tumor genes with your gene of choice. Replace all the tumor genes with resistance to caterpillars. There's our gene which causes resistance to caterpillars. And now you've got plants which are resistant to caterpillars. Replace that tumor gene with resistance to herbicides. Now you've got a plant that you can spray your crops with an herbicide. They aren't affected, but all the weeds die. And if you go along I-35, you take your car and you look around and you see the corn and the soybeans and the cotton, 98% of those crops are genetically engineered with those two genes. So believe it or not, regardless of what the activists say, it's a done deal. And it's in China and India, especially Brazil, Argentina, and those places, they are using this like crazy. And the pesticide usage has absolutely plummeted worldwide because of these. So it's really fantastic system. But here's another thing you can do with it, is in between the left and right border, you can put a whole virus sequence. Remember that virus sequence we saw before, the TMB? Let's go back to that one. Let's just pop this entire TMB sequence into this. And if you do that, then you can go ahead and add the Agrobacterium straight to a leaf, and every place the Agrobacterium touches the leaf, it transforms those cells, and you'll get the entire virus as a gene inside the chromosome. Then, when that gene transcribes, if you've got a 35S promoter from cauliflower mosaic virus, the virus pops out. Here I am, an RNA, and the lucky few that actually get exported to the cytoplasm, they go to town. Because all you need is one RNA, and the whole cell is doomed. The virus will multiply. And if you have a gene of interest placed in here, in the middle of this multiple cloning site, then that means that you're going to have a whole bunch of protein produced. So what does that look like? It looks like this, precisely. It looks like this. These leaves have been inoculated with Agrobacterium. And we have green fluorescent protein. And this picture is not lying. It's really bright. It produces a lot. Now I have a vector that produces, it's one of the top 3 vectors in the world. It's now patented here at Baylor University, got a patent for it. And it produces prodigious amounts of protein, of green fluorescent protein. It produces so much that I can put a UV light against the plants. We have the room lights off, and you can see, it's bright. You put the room lights on, you can still see it. It is incredibly bright. I can produce 40% of the total soluble protein in the cell is my GFP with my vector. So it's just really powerful way to produce it. And I bring this up because it's plant virus vectors, but you can use animal virus vectors to also deliver all sorts of things. So I wanted to get you into viral vectors now, because when we talk about vaccines, you can also deliver vaccines into humans this way, and especially into for veterinary vaccines as well. Okay? So those are plant viral vectors for protein expression. Any questions before we leave that? Okay, our next one we're going to look at then is fusion partners, and a better name for that, it's a better search term for that is affinity tags. Okay, somehow I got into cap locks, but anyway. Affinity tags, and we're going to go to, for this one, we're going to go to the wiki search for this, protein tag, Wikipedia. And what protein tags do, we don't have a picture for it so much. Well, let's go back here, actually, to these images so we can give you a picture of this. Protein tags let you do all sorts of stuff.